The delegates who have not participated for the poster session may kindly be seated, as in a short while we will be moving on to our next lecture, the evening lecture. They are requested to be kindly seated. everybody. Uh, as our speakers have already arrived, we are about to start the evening lecture session in a short while. The delegates are requested to be kindly seated.
Good evening, one and all. All the delegates are requested to take their seats for the evening lecture. We are going to start the evening lecture in a short while.
In this lesson, we're going to learn how to play the song See You Again by Wiz Khalifa. We're going to learn two parts to this.
All the delegates are requested to be kindly seated as we proceed for our first evening lecture of the 3rd ARRW Symposium 2018. A very good evening to one and all. May I now welcome the chairperson of the session, Padma Shri Dr. Ajay Parida, Director, Institute of Life Sciences, Bhubaneswar, and the speaker of today's evening lecture, Dr. Sapan Kumar Datta, Pro-Vice Chancellor, Vishwabharati University, West Bengal, to come on the dais. Please, sir. Thank you, sir. May I now request Dr. Himanshu Pathak, Director, ICR National Rice Research Institute, to introduce the chairperson of the session. Please, sir.
Good evening, everybody. You please take your seat because we are going to have a very, very important evening lecture which will be delivered by none other than Dr. Sapan Kumar Datta. Of course, a formal introduction of the speaker today will be delivered by our chairman, Dr. Ajay Parida, but before that, I shall introduce the chairman. Friends, it is indeed a great pleasure for me and I think for all of us that we have got Dr. Parida amongst us today to chair this important session. Dr. Parida is a very known person, particularly those who are working on rice biotechnology. But for my younger friends, for our research staff, Dr. Parida, with your kind permission, I would like to say a few words about you and your contributions. Dr. Parida is noted for his contributions in the fields of agriculture, plant molecular biology, and biotechnology. In 2014, Dr. Parida was awarded the Padma Shri Award by the President of India for his contribution in the field of science and technology. Dr. Parida is, of course, a man of the soil, born and raised in Odisha, is currently the director of the Institute of Life Sciences in Bhubaneswar, which is an autonomous institution of the Department of Biotechnology, Government of India. He has served as the executive director of the M.S. Swaminathan Research Foundation in Chennai during 2009 to 2017, so around 8 years or so. Dr. Parida's major scientific contribution is in the area of application of frontier technology for addressing major challenges in global and national declining agricultural productivity threatening due to climate change, sea level rise, and reduced precipitation. His research aims at developing location-specific crop varieties to cope with the adverse impact of climate change and has significant implications in bringing in stability and sustainability of the major farming systems. As expected, Dr. Parida has won several awards and recognition. Just to mention a few, he is the recipient of Umakant Sinha Memorial Award by the Indian Science Congress Association, B.M. Birla Science Prize by the Birla Science Foundation, National Bioscience Award for Career Development by the Government of India, Nashi Reliance Award for Application of Oriented Research by National Academy of Sciences, India, Tata Innovation Fellowship of the Department of Biotechnology, Government of India, and of course, several recognitions besides Fellowship of National Academy of Agricultural Sciences. Dr. Parida was elected as the President of the Agriculture, Science and Forestry section of the Indian Science Congress in 2012, and he was the President of Biological Sciences section of the National Academy of Sciences in India in 2014. Dr. Parida, these are just very few words about your contributions and the research which you are doing for the last several years. It is really an honor for all of us, particularly for the staff and students of this institute, National Rice Research Institute, to have you today evening to chair the session, and the session is going to be very important one, a very scintillating one, and as I told you in the beginning, a very renowned personality who is amongst us, Dr. Swapan Kumar Datta, is going to deliver the speak. Thank you very much, Dr. Parida, and also Dr. Datta for kindly accepting our invitation. And now I request the chairman of the session to please introduce the speaker. Dr. Parida, please.
Well, thank you, Dr. Pathak. I came here to listen to the lecture, and finally I landed to chair this session. And thank you, Dr. Pathak, for giving this particular honor. And I am at a loss to introduce Dr. Datta. He is a very good friend, a colleague, and more than that, he has also been a teacher to me. In fact, I spent my overseas associateship in ERI in his lab. So he has been a friend, mentor, guide, well-wisher, and whatnot. And Dr. Datta's scientific contribution, I think it is known to all of you. He is pioneer in rice research, specifically in the transgenic, and you name any transgenic in rice, his and his group has it. Whether it is for disease resistance, salinity tolerance, abiotic stress tolerance, or nutritional enhancement, many things. In fact, his laboratory is considered as a factory for producing transgenic rice, and many of the Indian researchers, specifically he had, when he was in ERI, he was, being an Indian, he has been promoting several of us to spend some time in his lab. My first interaction with Dr. Datta goes back to 1990s when I was in Swaminathan Research Foundation, and he came to Swaminathan Research Foundation for a conference to on biotechnology. But he insisted that I should go to Pondicherry, where MSSRF is having a bio village. Although I was thinking that a transgenic person, what he has to do with bio village, but I think that shows his keenness to take the technology to the poor. And how do we not this? I don't want to go to the GM debate, but if we would not have this controversy with the GM, I think the number of transgenics which would have come from or which would have been deployed in India, Dr. Datta and his group, specifically his wife, Karavi Datta, they would have led the nation in deployment of transgenic plants in the country. But so that shows, I'm not going to his work, whether it is in Zurich or for some time in ICGB, then ERI, then he was in Calcutta University. But most of his part also he spent as a great, agricultural leadership position, that is the Deputy Director General of ICR. And I have the privilege of discussing many of the issues on agriculture in his house in NASC campus, because his wife was not staying there, so we were using it as a bachelor's den, and all our discussions used to be there. And his scientific contribution has been quite outstanding. Just because all of you are scientific personnel, his citation index is now, his citation index now comes to 10,000 citations. And I don't think any of us will aspire to come to that stage with a H index of 42. I don't think I have to mention anything about his scientific contribution, and hardly anybody in this, in India, in agriculture field has that much of citation and H index. But more than that, he is a very simple and interactive human being. I don't think that we need an appointment when he was DDG to get to his room, but he will just, you can walk in and his doors are always open. So we have a scientist, a thinker, as well as a great human being with us to deliver the evening lecture. And he also served as the Vice Chancellor of Vishwabharati, which also brought in the cultural aspects to his scientific aspect. But I think the topic he has chosen is rice and politics. I think let's hear. I am sure that this will be an interesting lecture. And this topic you can give only when after retirement, not before retirement when you are in the job. So without telling anything else, I'll now a privilege to invite Professor Datta to speak to us, and I'm sure that we'll learn much more to what the rice research priorities or even the other agricultural priorities in the country. Welcome, Dr. Datta.
Thank you very much, Dr. Ajay Parida, for a very kind introduction. And it's nice to listen to my friend and colleague. And in fact, there are quite a large number of people are in the audience who are rice scientists, Dr. Vian Singh, Dr. Jena, and many others. And so I have been privileged that associated with many of those seniors as well as young people during their course of study as well as their interaction with me. Well, today morning we have heard Dr. Trilochan Mahapatra's, the DG ICR's presentation. That was a lovely presentation and well thought, well structured, and many of the issues that he raised, and he also touched lots of concerns, and those concerns probably he left to us to elaborate during our discussions and to bring it in the platform. Well, what I thought in my presentation, I will definitely touch a bit of rice science that mostly myself and my group were associated, and also partly the global science. Not many things that I have included, only I got 1 day time. In fact, even that time was also not enough. We were attending a very important meeting of Nobel laureate prize sort of summit. So very little time I had to put up those slides. So please excuse me if not enough what you expect from me to speak tonight. Rather, it would be selective, a few areas that I have chosen to bring it to you. And probably I would be happy to get some questions from you at the end of the session if the organizer, Dr. Himanshu Pathak, agree that can be taken up, or S.R. Das, Dr. S.R. Das, the president of the organization Arrow is sitting here. I also would like to thank very much the organizer for inviting me for this particular meeting. Well, we know that rice feeds 3 billion population, one of the most important food crop of the world. But the picture, what you see here, it looks like a very rich man diet and having every other aspect, and there is a little bit of rice. Now those here who have traveled Korea, Japan, and China, you know that normally these people eat rice at home with the main dish is rice, and additional things come one by one. But when you are a restaurant, a good restaurant or a very sort of professional dinner, rice comes at the end, a small bowl of rice. In Korea, it comes in a very special way, and you have to take rice from the bottom of the pots also, because they think the most nutritious part remains in that part of the container. But in this case, the picture, why I choose this picture, that what the diversity of the food as a nutrition component is required is everything is there. It's a balanced diet, and a small amount of rice is there to nourish your pleasure as a dinner or lunch. But that's not the case. Majority of the people who live on rice they would have the luxury to have this kind of mill. For that reason, rice need to be nutritious, rice need to be good quality, and particularly unlike China and Japan, they have a specific likings of rice, but in India, every state has its own type of rice, so those different regions, this rice improvement also need to be made. So it's again very different challenges for India to look into that particular aspect. Can you reduce a bit light? Otherwise the slides cannot be seen. Now the rice, I was talking to Dr. Chaudhary, Ram Chaudhary, is he here? Dr. Ram Chaudhary had a beautiful poster of Boro that grows in 4-degree temperature. At the same time, during harvest time, it can stand 40+ degrees centigrade. That is the rice needed. And I saw there is a poster that he put up here. Now, I was talking to him that rice, in fact, the ancestor came, say, 135 million years back. But actual record of rice, Indica and Japonica, it came around 8 to 10,000 years back. And our Atharva Ved and Rig Ved doesn't have any information, Bible doesn't have information, Egyptian inscription and also record does not show rice in the ancient time. Although the rice definitely originated from India and moved all through the different countries. And today, more than 115 countries, they grow rice and consume rice. So it's a quite interesting crop of the world. Now, even the Balinis, Indonesian legend, they say the Lord Vishnu, actually the god of the fertility and the water, gave birth the rice to the earth and asked to practice that rice for our people. This is the legend. And the interesting part of that, now if you see, there is a company, God Vishnu Rice Unit. Now if you go to the Google, if you want to look this particular information, you will find this company is coming more, that God Vishnu Rice Unit. So see, the science in one part, the evolutionary aspect, the scientific aspect, at the same time, you see how the trade and business comes also with those kind of names. And there are a lot of those stories all around the world is available. Now this is the picture, I took it myself in one of the museum of Taiwan, where it shows that around 7,000 years back, some fossil rice was recovered and they kept it in their museum, which is also a quite interesting part. Now, as we know that rice has very diverse ecosystem, that rain-fed upland, irrigated, lowland, and deep water, meaning that the rice can grow 2,500 meter high altitude, at the same time 6 meter deep water. And across the world, when a particular crop can have that sort of diversity, imagine how flexible this crop is. Now imagine that the genes present in the rice, or the genes that makes the genetic makeup of that particular crop, how that can be activated and deactivated and can make the phenotypic expression, at the same time allow the plant to survive under extreme conditions. So all possible genes that what we talk today for biotic and abiotic stress, nutrition, and many other aspects, all are present in rice, including genes for nitrogen fixation that also present in rice but remain in inactivated fashion. Now this is the things that the certain a glimpse of a picture that shows the different types of rice of the global rice that grows at ERI, and that shows the different kinds of traits that are available in the rice. So, which is very clear that whatever the designer rice that we need today, or we need to develop because of the adverse climatic condition, this rice material can be utilized as a donor material, and that can be utilized for any improvement of this particular rice. And there are certain genotypes that now being developed, and today morning that I learned that CR Dhan 310, 801, these are the new latest material that has come. One is high protein and one is submergence combined with the drought tolerance, which is very important component. As I told you that the genes present in rice that can be, if that can be utilized properly, all kinds of traits can be improved and the rice can be grown across the different ecosystem. Now, when we are living in the genomics era, definitely the one important aspect that we must decode the rice genome, and which has been done long back. And the question is, the fingerprinting, the genome sequencing, all of these things are now quite easy kind of tools. But using that material, ultimately the phenotyping or the utilizing in the pre-breeding as well as plant breeding is the challenging job, and which is still left. Now, everything what you see there is a sequence. Now, as earlier I mentioned about the different traits, now you see this is all traits are linked with the sequence. Now, if we now can combine both, so any improvement and whatever the unknown things we see, also we can connect it with the sequencing information. Now, with the genome map, as I just mentioned, one is that bioinformatics, that is definitely need to be connected. The gene discovery, the next step. The design, the crop design, that we need to design what we actually are looking for, what we want. And then using genetic engineering or marker-assisted breeding or other tools like genome editing nowadays is coming. Then ultimately we get improved products. Now, when we come to the regulatory process, the politics started. And other concerns, business and issues, one after another things started coming. I'm not going into that details of that particular aspect, but in course of my presentation, there are many, maybe a few things might come. And ultimately, the farmer's choice and use can be utilized for better rice development. Now, in one way, we're looking for genetic gain of the particular material. In other way, we are looking other disciplines, particularly soil and engineering section, that to combine with that how we can make mechanization and scaling up and get integrated approach of all these things together, we can harvest a better product. But now, even during Tagore's time, I mean Rabindranath Tagore, the Nobel laureate in literature, even he mentioned that if any year the farmers produce more rice, he will have some kind of problem of his fate because he will not get price. And today, the scenario did not change. If I have more production, less price. If there is less production, anyway, farmers don't get the price. So I think the problem remains same, and this need to be addressed. Although today, Dr. Mahapatra was mentioning that government new scheme that shows that to give more minimum support price, 50% more minimum support price to be added to the price. But then issue of the price and other things, I would come at the end of my talk, then we'll elaborate on that, how that can happen. Now, if you look this particular picture carefully, you will see that the planet has natural science, ecosystem, organism, either rice or any other crop. Then the basic science, cell, genes and molecule, etc., everything will come. The second part is policies, restrictions, trade, conditional, and institutions that would handle all of these things. That comes under the second part. And of course, the driving force is science and technology, and the issues that all linked with profit. Now, the third part, this would not come unless that has a societal and social impact. One is the social, society, community, household, individual. Again, as you know that with the human genome mapping, now the international scientific community are studying now, looking at the different genes that having the impact on our mind, how our mind works, how our mind reflects and conceives something. The people are also working on those genes. Even Japanese are working on some genes, particularly with the diabetic friendly rice, so that if that rice can be eaten by the people, they would have some stimulus that insulin would be produced, and it also mind driven. So I think all are connected today in science. That is a very interesting part, that and that's the reason I said that we are living now in the genomics era. Even our emotion can be also monitored and judged by the genes. And of course, there's a few people that are very much behind this entire scenario. The Darwin, Mendel, and Watson, Khurana, and 3 World Food Prize winner, Fraley, and then Mark Montagu, and Parek, what is this name of the lady? Clinton, no? Meridel Chilton, and Max Planck. I think I kept this picture, but in the Nobel laureate meet, there was a discussion came, what is the model of an institution, what is the model of a particular science that should drive and that should given the impact in the society? Is there any model? So people talked about Max Planck Institute, people talked about Friedrich Michel Institute, people could think about ETH, people can think about, say, MIT, Harvard, Princeton, Stanford, etc., etc. But these are the institutions of certain people, and today we are in the 3rd generation of the university, which are mostly driven by networking. So I think this is a very big differences and changes that are taking place. And this is all science and technology driven. And with that sort of things, that the products that are becoming or we asked today, that's the reason when I was at AIRI, and the setup of area of plant breeding, it is something that young people also would be interested to work. The same thing I have seen in Shifa Zhang's lab in China. In the morning time, all the students work in the field, they work for the breeding work, etc., etc. After that, at 12, they go for lunch. It's a 2 hours lunch. Sometimes it could be 3 hours. They take a little bit of nap. And then they come to the laboratory, and then again they work till midnight. So that means in one day they work 2 days' work even more efficiently. But what is the motivation that made Chinese people work so hard and then others cannot? I think this is the things that today people or the young people like to work in a kind of science-driven kind of something, need to motivate. So in the morning they do breeding work, which is also a bit of marker-assisted breeding or transgenic or some genomics. And then in the night they do the laboratory work. So all the combinations together that make big differences. But that was not the case when, earlier times in 19th or 20th century, or when Dr. Gurdev Khush and other people are working. It was not the scenario behind it. Here you see a few, the World Food Prize winner, the main contribution in rice, science and rice policy. Swaminathan, Chandler, Muhammad Yunus is a social economist. Henry Beecher and Gurdev Khush got together, and hybrid rice godfather or father of hybrid rice, Yuan Longping, and Monty Jones from Africa got the last Rice World Food Prize 2004. So for a long time, not many rice scientists are getting this prize. Anyway, but these are, their entire work is very different. I was just telling, when I was working at ETH Zurich, I requested Gurdev Khush to bring some material and to send some material. And in 2 weeks' time, Gurdev Khush was visiting ETH, and he gave me one full briefcase, and it's a heavy weight. I was surprised why he is giving the briefcase to me. So he said, Chopin, open it. So I opened it, and then I found all rice seeds, different packets of rice seeds he brought for me. All the elite germplasm and important, interesting material for genetic transformation. Now, that was possible, say, 15, 20 years back. It is not possible today. Even you try your best to fill up all forms and protocol, etc., etc., even FAO designated material sending from one place to another place is getting difficult. So now either I don't know whether I call it a politics or it's a policy or lack of vision or lack of policy decision. Either way, it is hurting the science to move on. It is rather getting difficult to work today when germplasm movement and any material movement is getting extremely difficult. And even once I made a statement that we have plenty of germplasm in the gene bank, what is the value of keeping germplasm in the gene bank if we don't use them? So it is better to collaborate, including private sector, on those materials to get genes and discover the genes and ultimately use those genes for a better improvement of rice. So the next day, newspaper put a headlines that Chopin Datta giving gene bank to private sector, and the gene bank would be now totally given to the private sector, and etc., etc., etc. Even if Ajay remember, someone wrote to Swaminathan also to check out with Chopin Datta whether Chopin Datta is aiming or arranging something like that. Of course, Swaminathan didn't believe that. He wrote a letter back to those people, activists, and also gave me a copy. Then, of course, I explained to Swaminathan. So I said that I still believe that it is useless if we do not use our germplasm. If any material today in the germplasm are sitting there, if the ecosystem is not suitable to grow them, do you expect that 50 years from now this material can be grown in our ecosystem? Rather, the genes present there, the genetic makeup present there, we must understand them, we must learn from them and use those genes and trade for improvement and to get a better crop development. And for that, any collaboration across the world need to be made so that we can continue doing science rather than spending money on just keeping gene bank and doing nothing. But anyway, I hope there will be no controversy again on that aspect that I am no longer attached with either NBPGR or ICAR. So that Norman Borlaug always had a very nice dream of thought that many of the germplasm, many of the material, either wheat or rice particularly it doesn't have a rust. So rice must have some resistance of rust, which can be used for wheat, when wheat is susceptible for rust. Similarly, many of those material can be utilized for rice improvement, what is present in other crop, which is he firmly believed that, and he used to talk about this very openly. One of the things that as the land, Mahapatra was mentioning about the decrease of land, I think that China did one wonderful work that they made their rice productivity very high. If you see the rice productivity in China, which is very high, average 6 ton per hectare, with India or indica rice, particularly the rice, because our ecosystem is very adverse. Under very constrained situation, our rice growers are growing rice. The optimum condition, of course, we can also have 6 to 7 ton per hectare quite easily, but as our entire area is not optimized to grow the high yielding rice, and that's the one thing China perfected their conditions to grow high yielding rice under optimum condition, which is missing in India. Probably in future we need to look, reduce the land, but only grow rice in an optimum condition, including soil, fertilizer, etc., other things together. This is AIS64. Just imagine how much combination is required. At the same time, if you add a gene or you want to improve a trait, there will be so many unintended or something changes will take place in that particular genome combinations. And then activists immediately will be jumping on it. Oh my gosh, so much of changes, so much of unintended aberrations, mutation, etc., etc. So that means all the biotech products or GM is bad. But if you look at this one, when you say it's conventional breeding, the same slide, basically same slide, this is acceptable. But when you made it in a colored and showed that what are the changes taking place, then it could be unacceptable. That means something definitely wrong. Well, but the conventional breeding or transgenic breeding, either way, the fact remains same. That is, after crossing the F1, that is a Mendelian or a non-Mendelian ratio 3:1, the product development. Now, if you go for the from gene discovery to transgene transformation, receptor binding, etc., etc., and you may get feedback inhibition, gene silencing, gene inactivation, unknown mutants, sterile plants, pleiotropic effect, and fertile plants. But ultimately, you want a homozygous material, which is true for conventional breeding, which is true for all kinds of plant breeding, either it's a mutation breeding, transgenic breeding, or any kind of breeding. If you want to improve a crop, you will definitely get all of these things here. But the plant breeders or the conventional breeding ignored them. Esar Das, I don't know how many of those things he might have seen in the field, he threw them out. He doesn't look, he doesn't want to or waste his time to look into this. Rather, he looks for, as Gurdev Kush always looks for the material, what is good, he just picks up and go for the next generation. And that is the way it is. But in the modern breeding, one can predict, one can detect, one can understand the signs. And that is the difference between this particular science and understanding the science. Now, with the architecture of the plant, before we come to the designer rice, today what everybody look for, but it is something that the 45-degree angle of the leaf phyllotaxy should be there so that it can absorb the best possible solar energy. And that is something very important, and lots of work earlier time physiologists did this kind of work and they found the result. At the same time, there are certain genes have been found. That gene shows that alteration of a particular genes or inactivation of certain genes, then it changes the length and all the internodes also accordingly, proportionately they got reduced. Even I have not published a paper, which probably sometimes I will do it when I get time. And we used one gene, and even I have all the data with me. That gene changes the architecture of that plant, the height of the plant, all internode length, even including individual seed length has changed. Everything proportionately reduced a little bit with change of one gene. So that's so powerful that these kind of things can happen. With the alteration of one gene, the entire architecture of that particular phenotype can be changed. So if it is intended, if it is desired, that means something can be utilized for further alteration and further designing of the material. Now, if you from here you come to this stage where you perfected your material, the phenotype, what you'd be looking, this one particularly, if you look that you have an angle of your leaves and the absorption of the light, etc., all perfected, as well as the height of the plant. And of course, a particular gene, that is D1 and others, that also, which is the main gene for the green revolution, and that reduce the apical dominance of the plant, resulting more tillers to come and dwarfing in nature, and that gives more yield. And that was the success story of the green revolution. And today, there are so many traditional tall varieties, and these tall varieties, farmers do not like to grow them because they are tall, they lodges very quickly. At the same time, they are not much high yield. So if those material, we could use those genes and alter them, reduce their height and make a higher yield, that could be beneficial to our need. Which is definitely, for a long time I have been promoting this particular area, and I think A.K. Singh probably did some work with the Kala Namak rice. Hopefully Govinda Bhog rice, is A.K. Singh is here? And then this can be utilized in the future. One GW2, that ring type protein ubiquitin ligase activity, could enhance the yield by enlarging the size of the seed, and that definitely would increase the yield. The GHD7, I think this is again an interesting work, came from as I mentioned that rice is so flexible, it can grow anywhere, everywhere. Now, in everywhere, the same gene present in the plant does not express. Now, in certain ecosystem, these genes got activated and expressed, and you got the expression. So interesting, and if you now can control that activation and inactivation of that gene, accordingly, whatever is required by us, this can be achieved. So such a wonderful and powerful this kind of scientific understanding. It was mentioned in the morning, emergence material that came from and there is material available in India. I hope there's lots of work is going on. Sharno Savan just came through this concept, and that being utilized and providing today many of these Savan material. One of the things that to the phosphorus deficient soil, Kaushala material, that the donor material that gave that gene, and that gene now being utilized. A few trees in India as well as elsewhere working is the work initiated at NRRI, and particularly basically in Japan, and then it continued. And those of the work definitely, when phosphorus deficient soil and phosphorus is expensive, in both ways this work can be very much utilized. I kept this slide, and now I'm glad that Dr. Chaudhary is here. Where is Chaudhary? Ram Chaudhary left? This man went in India to NRRI. Ram Chaudhary is the person who did that. And then this material went to NRRI, and Gurdush, this material, and Ram Chaudhary informed him this. There are certain material at NRRI. At that time, the name was NRRI. Material grows here. It material right attack this plant. So Ram Chaudhary didn't know that what is all about this material, but he found some interesting result of this material. So he told to Ghosh, and Gurudev Ghosh asked him to get this material. So he gave it to ERI. Now while Gurudev Khush, first thing he did, he crossed this material with a susceptible variety, and he found here segregation showed Mendelian segregation 3:1, that means one dominant gene is involved, and ultimately after studying he named the gene XA21. Now the Cornell group came and took this material and patented it, and then started different kind of stories. And at one point when we developed XA21 enriched many rice variety, including AIS2018. So we wanted to give this material. We started giving this material. We gave to India and different places of Philippines, China also. But then there was this issue came that we should protect this material and we should charge for it. But ultimately it didn't happen, fortunately. But unfortunately, there was a wrong notion was taken up that certain proprietary rights, certain business did not take place. And I believe that at ERI, the mission and vision of ERI is to develop the material and give it freely to the developing country for all purposes, for research as well as for utilization purposes. I believe ERI is still probably having the same vision and mission, and Dr. Jena is here, he might be able to confirm that. And that's the story of XA21, wonderful story. Now a lot of pyramiding has taken place. XA4, 13, 21, 28, etc. China also, Shifa Zhang's lab also doing some more work on that particular aspect. The climate change, I think there are a lot of experts are here. Himanshu Pathak is one of them. Definitely one of the issues that if we really get more CO2, that might disrupt our metabolic pathway of plant protection, which could be dangerous, and we should be prepared for that. And definitely it would adversely affect the yield, and unfortunately, but again, if the material staying in the gene bank, it doesn't come, it did not help us. And well, I was at ICAR, my good friend Kailash Bansal utilized huge germplasm of wheat and grew them in different places of India and found lots of interesting material, and that showed lots of different character. Now those of the germplasm need to be utilized for future pre-breeding as well as for future source of the material. But I don't know at the moment what's happening. Bansal retired, and now someone else need to take up that kind of work. This is a hard work. It's a very hard work to look or phenotyping the material. Genome sequencing much easier, believe me. You don't do anything. You give the DNA to some company does the work. Even annotation could be done by others, and so most of the work done and information is available to you. If you have the money, and you can direct the funding, and this is done. But phenotyping is a difficult job, hard work, and that need to be done. One of the things that drought tolerance, there are many genes are available. I think this is an interesting area. But to me, the most interesting part, this one, the genes, the transgenic material, the genes could close the stomatal aperture where the control does not have it. And closing the stomatal aperture allow the plant not to transfer the water, and that's the way it protects the plant. In the night time, stomata aperture opened and it works. Now, I don't have the slide with me. Aninda Bandopadhyay from ERI, now he's joining Syngenta in China, and he developed a material with the genome editing where this control or regulation of stomatal aperture and stomatal number can be regulated, which is, that means genome editing, the CRISPR-Cas9 is a possibility that some of the reshuffling of the genes can take place, and some of the knockout of the genes or some of the modification can give certain phenotypes which is desirable. Pusha Basmati 11, that was elaborately in the morning, as well as AIS10, the one hybrid, that basmati type hybrid rice developed by NRRI, which is a tremendous important. But again, either politics or trust, whatever it is, when NRRI developed this material and gave it to the different companies, company took that material. There is an agreement that under this agreement, they have to come back and pay some royalty, etc., etc. Once they got the material, thank you very much. Rice is for everybody. You develop the rice with the public funding money. You gave us this rice. We got this rice. Now we will use it. We do not need to go back and to return back anything to you. This is very unfortunate part of our country, that we were losing the trust between the donors and the recipient, and if we cannot build up that trust, and this is the reason hybrid rice didn't go much from India, and where we have a tremendous potential and we could do much more, but that's sort of question and need address how to build up that kind of trust between the two groups. Now here is many different issues, but I would not elaborate. Just quickly I will go through it. The pesticides versus built-in plant protection, just like BT rice or sheath blight resistant rice, bacterial blight resistant rice, or any biotic stress tolerant rice would be definitely a preferred choice rather than using pesticide. The pesticides residues versus antifungal gene products. So some questions comes from the activist. Obviously, pesticide residues are not preferable rather than antifungal gene products. Benefit of green revolution versus traditional rice culture and practice. I mentioned in the beginning of the tall variety versus dwarf varieties. Improved varieties do not last long due to the breakdown of resistance. Sometimes certain things doesn't work after a few years, and immediately people jump on it, particularly activists jump on it. Oh, this technology broken down. This technology doesn't work. But even for any varieties that developed by independent of any other method, it will break down depending on how much you use it in the given place. And that's the reason breeding is a continuous process. Continuously you need to breed new material and bring the new material. A bridge of diverse eco-friendly microflora adapted with improved cultivars may work well for sustainable agriculture. This is a very important component. Those are working with microbiology and ecosystem. Continuous monitoring of deployment of improved crops in environment is essential for longer nutrient-rich soil, fertility and environment-friendly, higher productivity, which is long-term research. Lots of people are working. Now, biological nitrogen fixation. As I told that rice do have these genes. In course of evolution or in course of practice, we are adding more fertilizers. Definitely, whatever reasons, these genes got inactivated and it doesn't function anymore. With the given right promoter to induce those genes, to activate those genes, I'm sure with the correct type of association with the rhizobacteria, this nitrogen fixation can be achieved. I think I'm sure someday this work would be successfully carried out and we will see the successful result. Same thing with the C4 rice, which is the reality, but unless we put enough effort and energy and work, we cannot expect that this thing will be done just very quickly. Nutrition, definitely, iron, lysine, and provitamin, and the high protein rice now, NRRI has developed it. I think this material can be combined with provitamin A and high iron rice. That should be a wonderful product development. Otherwise, that European, or maybe in India, if we do not use or don't want to use GM or biotech products, and as we can make certain other way, certain variety development, certain product development, including vacuum packed brown rice, In this case, it is a vacuum-packed brown rice. Italy, they use it. But the problem is they use so much of small amount of rice. For them, it's a luxury to use or not to use it. But this is not the case in India. We eat rice every day. If one day we don't eat rice, those are rice-consuming people, they would feel very uncomfortable. They need rice. So it may not be the same, but nevertheless, one could diversify the way of using. Of course, the golden rice, that's one product developed long back, but it didn't go anywhere. I think here, science, some people want to do science democratically, or this democracy is something some activists, they design their democracy. If they are very active, that's the democracy, that's the way they want to control it. Most of the scientists, they keep quiet because they are not involved in the work, so they think why to put their finger in this particular work. A result is that even good science remain in the shell and does not go anywhere. Even the carotenoids, if it's degraded, if we keep it for a longer period of time under open room with the sunlight, so we also knock out the lipoxygenase gene, and then downregulation of lipoxygenase gene also can reduce degradation of carotenoids. That also we published. So we continuously doing different kinds of research on golden rice and high iron rice, low phytate rice. There are several such publications came in last few years. But I do not know that this particular work, except publication, except doing science and enjoying science, how much that would have a benefit to the society or benefit to the impact of utilizing. Even then, even the golden rice, we also developed dihaploid golden rice. This is also published in Euphytic as well as PLOS ONE. You can see very clearly the homozygous material. You see that this is DH line, this is a haploid line. So even there are certain ambiguity that material IRI received from ERI that has some problem with the carotenoids expression. If they could, I suggested them, I don't know that they have done it, but in our material, the material we developed, we developed dihaploid material at Calcutta University. This material is with us, so that uniform material can be utilized whenever there is a time for it. And even then, if we look at haploid stomata, diploid, dihaploid stomata, as well as tetraploid stomata, there's a clear differences that, so from physiology point of view, from genetics point of view, from anatomical features point of view, everything very suggestive and very confirmatory. The mineralization of the minerals that taking place in the rice seed is very important. One of my students, Martha, after completion of her work on high iron content, she further continued that work. And then, of course, there are more such information and publications are available, but I did not keep them. And other part is mRNA regulation, nutrient homeostasis in plants. There's wonderful stories. And we found a lot of transporter genes that involved in rice that transporting this iron. And at the same time, it also transports many other metals. So it's an important thing that we need to regulate. We need to understand that particular science and regulate it, which one we want to mobilize or to express in the endosperm and where it should be protected. It should not go there. Or the simple thing that as golden rice or high iron rice, we used a particular promoter, endosperm specific promoter, to express it in the tissue specific manner, and that also can be carried out. 1865, 2001, 2018 today. It's a long story of genetics, and we have to believe this science, and this science is working. Of course, epigenetics and other things are coming up, which is also a new challenges. Of course, this is the way science always new, always young baby, because all the time you learn something new and something development will come. And that's the thing, it is exciting. It can never be depressing to do science or to involve in science. These are one slide that shows that bacterial blight, Bt rise, Bt development. Here you see the drought tolerance, herbicide tolerance, etc., etc. So major traits probably we could introduce and improve in rice, and we did that, and it works. And I strongly believe that this is such a powerful science that if you have a right design and right system of work, and it works. Now the next generation of genetics in plants, I think the challenging work. That is not the genome sequencing, it is not annotations, it is the phenotyping, which is a very hard work. And now this is the part that one need to see, that what combination you would like to have your marker together, and locations, etc., etc. But ultimately, the combination you can design in the laboratory, and ultimately you have to use it in your like phenomics facilities or greenhouse conditions. Then, as you do not have luxury now to do the breeding as Gurdev Khush and others did, or Norman Borlaug did, but today we work in a laboratory scale, but then that can be scaled up with the molecular breeding work, and ultimately material can be utilized. Here's just a part of the trade issue, that the market value already, what is going on is 13.2 billion, but actually it's 160 billion of few crop, not rice only. Rice alone is $200 billion. Now today, rice globally produced adequate to some extent, as because still quite a large number of people don't consume or don't have access to rice. But at a certain point, I believe that this market of rice would be more regulated, and in that case people can see the value of the rice and the amount that involved in rice. So those company or those scientists who have a product of rice, particularly on biotech, and that would give them dividend in future, and that has to come. Now this is the slide I was just mentioning, that Shingur is a very known story today in Bengal, because nano car factory Tata wanted to build up, and that was not possible because the opposition political party opposed the government not to have this nano factory, etc., etc. But the fact is, I'm not going to that politics. The fact is, 15,000 people live on only 997 acres of fertile land. So small holding farms, and such a huge number of people live on that particular land, small amount of land. Now, as Prime Minister is looking for doubling our farm productivity or farmers' income, if our farmers, large number, live on such a small amount of land, no matter how good science you do it, no matter how good management you do it, it is not possible to have a better income generation from that limited land resources. Now, what are the possibilities? The consolidation of land. Even story I can tell, even Tagore himself has a story. One time he was going in a village during his Jamindari time, and then he was rested in one farmer's house. And then the old man, not very old, but a farmer, he requested Tagore if Tagore could give a job to his son, because Tagore has Vishwabharati, Tagore has a lot of things. So Tagore was surprised. Tagore thought agriculture, farming is a very attractive, and your own land you are cultivating, etc., etc. And Tagore was surprised and said, why should your son get a job, will get some small amount of money? But the farmer said that the whole family depend on this particular land, particular crop we grow on agriculture, but our profit is not enough to meet our requirements. So we need one son get job. The real tragedy is today farmers even looking, more and more farmers are looking for a 5,000 rupees job or even less to give up the agriculture in many areas. Now, given that scenario, and if you want to double the income of the farmers, how that is possible? Unless we plan a good number or a proportionate number of non-productive farming land, or farmers need to move out of agriculture and engage in other work so that they generate income. What China did 20, 30 years back, they keep on agriculture, simultaneously large number of people also engaged in other work so that they gain some more economic benefit. And that way the transition started long back, and eventually it all set today. So only productive land for each crop are utilized in China for having high yield, which is not the case. That means planning is still missing, and we have not yet planned that optimization of all other or integration of everything together, that we need to use the high productivity. Anyway, I would not go more details on that. And this is the story that I thought I would bring it to you. As you know that this is a slide I often showed to the people, that North versus South. We are in the South, huge number of people, and not utilizing management, not utilizing the technology, etc., etc. And large number of people depend on very small thing, which need to be changed. If you don't change it, only science would not do enough for our rice improvement. Now, last few things that I would like to elaborate before I end. That is, rice price increased through last 3 decades, adjusting the inflation. That means our price did not increase that much in domestic as compared to international, if you look the inflation. This has an impact on farmers' economic benefit. Higher price benefit to the farmers and investment, but difficult for the poor consumers, because 30% of their income goes for the rice purchase. So it's a contradiction for them. Western diet culture impacted on low rice consumption, Japan, Korea, etc., also coming in India. So nowadays, more and more people eating less and less rice, but depending on some other junk food and other food, which is not good. My last but one slide will tell you. Rice is beneficiary to multi-purpose objectives that we must increase and attend to do that. The culture, economy, soil sustainability, food diversity, flood protection, etc. There are many things rice is doing for us. It's not just culture, it's in every other aspect. China today is a net importer and India is a net exporter country. We should not be happy for that. China is importing because it's a low price, that's the reason they're importing. They're exporting high value crop, so they're gaining much more than India. India, as Mahapatra told in the morning, we are not only exporting rice, we are exporting water, we are exporting human labor, etc., but with a very small amount of money. So we need to think about it, how we can optimize our need. India's can you put it in the, okay, India's food bill Act helps ordinary people. We get 2 to 3 rupees rice, kg rice, and many state government also even reducing 1 rupee more. That it's good for election. But it's not good for our economic benefit of the country, because it's at the cost of the development of the country, which eventually could benefit to the farmers, the infrastructure and every other aspect. because the rice cost is 27 rupees a kg. And then wherever there is absolute needed, the real below poverty line people, we can provide the rice free. That's of course, it's a good gesture from the government, good policy from the government, but not as a larger scale to basically at the cost of the country's development. Must have long-standing practical policy incentivization to all sectors, because today even the industry are not very much interested in rice or in producing more rice, etc., because it doesn't fetch the right price. But eventually it may, when there will be 10% less production of rice in any given year, that would be a disaster for India and then disaster for the whole world, because no one can feed India. India has to produce more rice for its own requirement. The last sentence is very important. Rice has a great social value, suitable eco-friendly food crop for India. Even the water use and etc., and it must get due attention with respect. So we must do every possible things to keep our rice production more, never less. We need to improve the productivity of the rice. We need to find a better market strategy for our rice growers. We need to value addition to our rice so that the farmers get a better price for their production. This is the government policy, need to look at it. Now the other things, what we have a serious problem, I'm sure NRRI and university and everywhere, everybody is facing or will face the problem. Today's regulation of any purchase or any utilization certificate, utilization or purchasing anything or doing science, you need to get a lot of stuff, a lot of equipments. You have so much of problem today to do that, you would not be able to utilize your funds in due time. So that's the reason funds are going to the international funding agency like NRRI. NRRI is getting or will get more and more funds than national program, because for NRRI or CGIR, it's easy, one-page utilization certificate, they will give it at a given time. No one will ask them how they have utilized the funds, how they have, whether they followed all government norms. But the government institutions, including NRRI, has to follow all government regulations, including university. So university institution will suffer, they would not be able to do the work, the money will go to somewhere else, and someone else would utilize the money, asking you to do the work for them. So this is a serious concern that I have, and this needs to be seen. Now, we need to be smart enough. I don't believe that Himanshu is less smarter than Dr. Jena, so that Himanshu has to find how to utilize Jena rather than Jena utilize Himanshu. So these are the things we need to look at these politics in a different way, so that all stakeholders survive well, they do science together. We enjoy doing science together. We want to collaborate doing work together, etc., etc. But need to be smart enough, not that I'm not utilizing the money, so I cannot give utilization certificate, someone else does it because someone is not questionable. And that sort of things we must change. This is my last slide. As I always believe that science is always new, and that is the excitement. Doing science, listening science, and engaged in science, always interesting. Thus I put Tagore's poems here. Whenever you are young, that always you are blessed with the sun, or blessings from the sun god, and that is Tagore's wishing for the young people. So the science does not have the boundaries, so the best possible science along with modern practice and management should be available to our young minds. So politics or policy cripples and any other things should not hamper, should not be the barrier for our young minds to nurture and to move on with science. Thank you very much.
Thank you, Dr. Datta, for the exciting lecture. When you are in love with rice and you are asked to speak on rice, I don't think you can stop or control yourself. I think that is the bottom line. I think all of you are waiting for the cultural event, but if you have some one or two queries or clarification from Dr. Datta, yeah. You have a mic? Yeah. You have a hand mic?
Yeah, thank you, Dr. Datta, for your nice presentation. I had only two information. One thing regarding the Orija longis terminata or Orija, but since Dr. Chaudhary is not here, I thought that.
He has come back.
Oh, he has come back. Anyway, so maybe he can tell better. But on your behalf, can I tell, and then you can add it? Yes. This Orija longis terminata, actually it was published by Dr. Devdutt from CRRI. He first reported that this having a resistance to all the pathotypes of the BLB occurring in India. At that time, he published a paper in the Current Science by Orija Barthai. The material, when Dr. Chaudhary was visiting CRRI, he took it to Patna. From Patna, it was taken by Dr. Khush as a hand-carried material to IRI. It does not have any accession number till last date when, even I have seen the Darshan Brar paper, there is no accession number given because it was a hand carry. And then the others we have told. So that material belonged to, and the work done was by Dr. Devdutt at CRRI Cuttack. So that's the information I have.
You are absolutely right.
Yes.
Any other questions or comments?
Just a comment, another one.
Yeah.
Okay, just
Ashwini.
one minute. When we compare the yield level, comes to India and the China. In Indian subcontinent, the yield is reported as a milled rice. In China, it is reported as a paddy. So these comparisons are not good because even in the member of parliament in our Delhi, he said China's yield is 4 times high. So I think we have to give a proper...
Dr. Bien Singh is absolutely right. I think we might need to change our yield component. Also, the productivity per day, per unit time, that's also another things need to be given.
Yeah, but
otherwise, we often got this question and in parliament as well as I have given many times these explanations, but we have not corrected it in our presentation.
You have to change it, otherwise we are getting
this entire things has to be changed.
Yes.
No,
I think Himanshu Pathak need to do these changes.
No, even if you do that, still it will be higher.
Sir, yeah, you are right, but in parliament we have reported several times, and now Indian MPs understand the difference, but in many papers it still comes with paddy and rice, and then people get confused, and then 4 times, 5 times multiplication.
No, Dr. Saab, I think whether it is 4 times or 5 times, yield is higher. Whether it is 2 times, but yield is higher. I mean that you have to agree. Yeah, that you have to agree.
You are right, but our yield, when we present in average, that average includes upland, which will be producing less than 2 tons,
that is the problem.
deep water, which is producing less than 1 ton, and then you put everything together, and then we say it is simply, and then we again reporting in terms of rice, so everything gets complicated.
Yeah, sure. Ashwini.
Thank you very much for the nice presentation. We really enjoyed it. I'm just curious to know about the status of golden rice. Would you like to give an update? What is it and where are we stuck? Because that is, I think, everybody is looking forward.
The problem is I'm on with the mic and in the television or some sort of a camera.
So this is
but anyway, I don't mind to speak the truth.
Thank you.
I think too much politics in golden rice and caused this problem. And Ingo Potricus, Peter Bayer, they made it very clear it should be IP free. It should not have any attachment of any regulatory matters in terms of transfer. But one wrong thing they did, that it should not be given someone having 10,000 income sort of. It should have been not given in that attachment. Now, certain company as well as some international organization, they got involved in golden rice management, and probably they want to regulate in their own style, and so the material did not move freely. What was ERI's vision and mission before, or what I believe today, even I always answer this that way, and I always, whenever I was working at ERI, I always tried to provide the material, whoever wanted, but on writing. But this control regulation ultimately affected this material did not move freely. And the people who got the material also has some conditions and restrictions. Science you cannot do with that kind of restrictions and conditions. Wherever science developed the things what I developed, or we could publish the papers, we have a complete freedom of utilizing those genes and material. But this restriction ultimately paid the price, meaning that now it doesn't go anywhere. And then certain material even utilized for field evaluation and even human consumption without proper sort of permission, because activists are very much behind it. They're all the time looking for something to find mistakes. So instead of science to move on, rather more and more questions and controversy started coming up. Ultimately, the material is stuck. And so rather, I still believe that this is fantastic science, and this science should have been given, should move freely. ERI, Syngenta, or anyone who has any restriction on this material, they should remove this restriction. They should make this material free. Ingo Porticus himself in India, he declared that this material is free from their side, so this material must move freely. Anywhere, whoever want to use this material and do science and do whatever they like to do it. But this restriction need to be removed.
Okay.
Thank you.
I think there are two questions are coming on the screen. There are two questions coming on the screen. The first question is, does transgenic rice really have the future in India? I think that future can only tell. That we can leave it to that. That future can tell. The second question, Dada, you can answer. That is XA.
Is there any future information on pyramiding XA38 with others? I think yes, definitely the work going on. I think today AK Singh and others working, Chinese also working on XA38, and also in India. The name may be different in China as well as in India, but this pyramiding work with XA38 along with 21 and others are going on. I'm sure at least in bacterial blight this would be a wonderful work when the entire pyramided material would be available and would be demonstrated in the field. And I strongly believe that transgenic crops including rice definitely have a future. This is science. If that doesn't have a future, science doesn't have a future. So science always have a future, and it should be utilized and should come. Thank you.
Thank you very much. I think our cultural troupe has arrived. That gives an indication that we have to close this session now. Once again, on behalf of all of you, let me thank Dr. Datta for this exciting evening lecture. Thank you very much.
Thank you, sir, for your valuable remarks. May I now request Dr. Himanshu Pathak, Director, ICR National Rice Research Institute, to kindly do the honors of felicitating Dr. Parida Padma Shri, the chairperson of the session, with a memento. Please, sir. Sir, I request Dr. Pathak to felicitate the speaker and the chairperson of the session, Dr. S.K. Datta, and Parida sir with a memento. Thank you, sir. Now, as we conclude today's evening lecture, I, on behalf of ICR, National Rice Research Institute and Association of Rice Research Workers, take the privilege of conveying my sincere thanks to the chairman and the speaker of today's session for sharing their kind views with us. I would also like to thank the reporters for the session, Dr. Sanghamitra Samantrai Madam and Dr. Umakant. I'd also take the opportunity to thank the audience for their kind attention and patient hearing. Thank you, one and all. After a day-long discussion on different aspects of rice science, it is now time to enjoy, and so I welcome the delegates to join the cultural fest to be held in the same venue in a short while. Thank you and a very good evening and night.